All right, so just kind of a summary of where we've been and where we're going. So if you remember in glucose gets converted to 2 pyruvate through glycolysis. You make 2 ATP, and you also make 2 NADHs. Those 2 pyruvate further get oxidized down to 2 acetyl CoAs plus those 2 CO2s. When that happens, you make 2 NADHs from the 2 acetyl CoAs that you oxidized. The acetyl CoA will go into the citric acid cycle, which we'll talk about in a second. That citric acid cycle will pump out 2 ATP. And also, the citric acid cycle will pump out 6 NADHs plus 2 FADH2s. And so what happens is the NADHs and the FADH2s will go into the electron transport chain to make additional ATP. So you'll end up with about 30 or 32 ATP, and I'll talk about that later. So we're going to move on to talking about the citric acid cycle and the electron transport chain later. All right, so the citric acid cycle is going to start the stage 3. It operates under aerobic conditions only, as I mentioned. So if you don't have oxygen around, you're just going to end up making lactate. It's named for the 6-carbon citrate ion of citric acid formed in the first reaction. Sometimes it's also known as the tricarboxylic acid TCA cycle or the Krebs cycle. I might refer to it as the Krebs cycle because that's how I learned it. Krebs was the guy that figured out what this cycle was. So just so you know. And I believe there are 8 different steps. So we're going to start with the first step. Again, you don't need to know every little detail about each individual steps. I'll go through at the end the main things I want you to know. So we'll start here. So in the first step, in this cycle, there's a molecule oxaloacetate that's in the cycle that gets produced in the last step of it. And so because it's a cycle, acetyl-CoA will come and bind with oxaloacetate. And in this process, you free coenzyme A over here. So the coenzyme A can go combine with more pyruvate to make acetyl-CoA. And your oxaloacetate will get converted to citrate through citrate synthase. So this is why it's called the citric acid cycle. So you can see this group right here gets added to this carbon right there. And now it's sticking above. So now you went from 4-carbon oxaloacetate, a 2-carbon acetyl-CoA, to make a 6-carbon citrate. So in the second step, you're going to do an isomerization reaction to end up making isocitrate. So you take citrate, which we just made, which is a tertiary alcohol. Use this enzyme called right here. And what that does is it does a dehydration. So this OH and this H are going to get expelled as water. But then in the next step, you re-add the water using the enzyme to move the OH right here. So the OH went from this carbon down to this carbon. So the reaction is just an isomerase reaction. So in this third reaction, it's actually going to be an oxidation reaction as well as a decarboxylation reaction. So in this, you take citrate to alpha ketoglutarate. So you're actually going to lose one of the carbons as CO2 right here. One of the hydroxyls is going to get oxidized to a carbonyl. And when one of those hydroxyls is oxidized, the NAD plus is reduced to NADH. And again, that NADH is going to contain energy in the form of electrons that we're going to use for the electron transport chain later. So you can see this group right here will end up becoming CO2. Some of those electrons get transferred to NAD plus to make NADH, and we made that NADH as an energy-containing molecule. In the fourth reaction, you're going to take alpha ketoglutarate and you can convert that to succinyl-CoA. So our CoA is actually going to show back up. Another carbon is removed as CO2. and it produces this 4-carbon compound from a 5-carbon compound that bound to CoA. And nicely, another NAD plus is made to make NADH. Again, we want those NADHs so we can use those in the electron transport chain. So how this reaction works is you're going to lose this COO minus. You're going to make CO2. This coenzyme A is going to get attached to this carbon with the sulfur. So it ends up down here. And while this is happening, your NAD plus is getting reduced by getting electrons to make that energy-rich molecule NAD plus. So now that we have our succinyl-CoA, it's actually going to get the CoA off again. So succinyl-CoA goes to this molecule called succinate. And how this works is you hydrolyze the thioester bond to remove CoA. And when that happens, it actually provides energy to add a phosphate to GDP to make GTP, which is a high energy compound. So it's very similar to ATP, and well, actually GTP actually makes ATP. It's guanosine triphosphate instead of adenosine triphosphate. And so how it works is you lose this CoA down here. You make it a COO minus instead of just right here. And in the process, the energy that you used ends up making GTP. And that's very important because it's another way essentially to make ATP. It's not a big deal, but the GTP that you form just gets converted in ATP. So ADP grabs a phosphate from GTP to make ATP, and then you make GDP. So it just exchanges a phosphate. So ADP takes a phosphate from GDP to make your ATP because the body's more used to using ATP instead of GTP. All right, so then in the 6th step, we take that succinate we just made to fumarate. You oxidize the succinate to make a CC double bond. And in this, we actually use another coenzyme that's kind of similar to NAD+. You can kind of think of it, it does similar things. It's reduced to FADH2. The big difference is the FAD is usually involved in making double bonds, whereas the NAD+ is usually dealing with carbonyls and those sort of things. So it uses an enzyme called succinate dehydrogenase. What happens is you take off one hydrogen from each of these carbons, you add them to the FAD, and in the process you're also adding electrons, and it makes FADH2, which we'll talk about now. So an FADH2 is just another one of these energy-containing molecules that will be used in the electron transport chain. So FAD is flavin adenine dinucleotide. It contains an ADP plus riboflavin, which is vitamin B2, which is why you need to eat all your vitamins. And this is the one that forms those CC double bonds. So NAD plus is dealing with carbonyls. FAD is dealing with CC double bonds. So you can see down here we have our ADP. You can see the part coming from the riboflavin. There and over here. And where the H's actually react on the molecule is you add an H to that end and that end. And when you add the H, you're also adding electrons. And those electrons are really what are going to have the energy that we're going to use when we get to the electron transport chain. All right, so we're almost done with the cycle. We just need to remake our oxaloacetate. So fumarate gets transferred into malate. You just add water to the double bond using this enzyme called fumarase. So you're just hydrating the double bond. If you remember that reaction way back from like chapter 12. So water gets added to the double bond. It's nice that it doesn't really matter what carbon you add to because it's symmetrical. So that's nice. And then lastly, the malate's going to go back to oxaloacetate, which if you remember, the oxaloacetate's what combined with our acetyl-CoA. So malate gets converted into oxaloacetate. You oxidize this hydroxyl right here to make the ketone, and in the process you reduce NAD+. So that NAD plus, again, it's gaining electrons to make NADH. And again, the oxyl acetate is regenerated, so you can keep running this cycle. And so just in summary, and this is mainly what you need to know. You don't need to know the nitty-gritty details of every single step. If you want to learn, obviously go ahead. But in the citric acid cycle, you add one acetyl-CoA. Each of the carbons in acetyl-CoA gets removed as 2 CO2s. You have 4 oxidations. You make 3 NADHs that will get used in the electron transport chain, and 1 FADH2 that will also get used in the electron transport chain. And then lastly, you actually make 1 molecule of ATP from GTP. But usually it's just counted as ATP because the GTP easily gets converted into ATP. So these on this slide is the main things you really want to make sure you know about the citric acid cycle for the test and all those sort of things. But luckily for you, you can look things up. All right, so briefly just going to talk about the regulation of the citric acid cycle. It's regulated at reactions 3 and 4, but honestly, just like everything else, all the regulations we talked about before, if you have high levels of ADP, that's what activates the enzyme because you want to make more ATP. If you have high levels of either ATP or NADH, it inhibits those enzymes. It's also true the step before that for acetyl-CoA. Again, pretty much all the enzymes, when they get regulated, if you have high levels of energy-containing molecules, you don't need to make them, so the processes slow down. But if you have low levels of ATP, you need to make more, so it will cause the pathway to keep going. All right, so here's just kind of a big picture summary of all. It's kind of complicated. If you had a meaner instructor, they might make you to draw this whole thing by memory, but we're not going to do that, so you're lucky. But maybe take a biochemistry class, they'll have you do that. But it just kind of summarizes all the things we talked about. Again, you want to see where you make those NADHs over here. We make our 1 ATP down here, and we make our FADH2 over here. And again, the carbons that came from right here are being extruded as those 2 CO2s, if I can find the other one. And again, it's a cycle, so those are the main things you want to know. And then also realize because each glucose molecule makes 2 acetyl-CoAs, for each glucose molecule, if you have to keep up with the math, you're turning this twice. So for each glucose, you're actually making 6 NADHs. All right, so in the Krebs cycle and glycolysis and the things we talked about so far, you broke down these sugars to make ATP as well as NADH and FADH2. And so now we're going to use the NADH and the FADH2 to give energy to our electron transport chain to drive the synthesis of even more ATP, which I've been alluding to. Again, there's a video if you would like to watch it. All right, so the electron transport, it's the same, you're transporting electrons. It's a series of reactions that happen in the mitochondria. So the citric acid cycle is already in the mitochondria. And so since the NADHs and the FADHs too, they're already there, they're going to react. And so what happens is the NADH and the FADH2, they're oxidized, and they make hydrogen ions and electrons. And so what's going to happen is these electrons are going to get transported to these proteins in the membrane, and these hydrogen ions are going to get pumped to make this gradient that will actually end up driving the formation of ATP. But we'll go over all that now. So in the mitochondria, in the inner membrane, not the outer membrane, the inner membrane, there's various protein complexes in the inner mitochondrial membrane. So these accept protons and electrons from the reduced NADH and FADH2. We'll talk about them more in detail, but this first complex will accept the electrons from NADH, whereas this, I believe the second complex accepts them from FADH2. And we'll talk about these more in a second, because it's kind of easier to see on the next slides. So in this first complex, like I was mentioning, this is where the electron transport begins. The NADH gets oxidized to NAD+, so you can see our NADH is now moving back to NAD+. and you're losing a hydrogen ion. And what happens is those electrons and the H plus are going to be transferred to the complex. And the electrons will actually drive, the energy from the electrons will actually drive to push these H plus in this inner membrane space. And that will be important in a second when we talk to it. So every NAD plus produces 4 H plus and 2 electrons. So again, the oxidation of NAD plus, it produces 4H plus and 2 electrons. Electrons are transferred from this first complex down to this CoQ. And then they're transferred, they're transferred to there. And then when they're transferred, again, these pumps pump electrons into the inner membrane space. So as the electron's going down, when it gets to 3, it also pumps more hydrogen ions across. So again, the main thing to know is NADH+, it gives the electrons to this first carrier. Because the electrons can go down this chain into the complex 3 and then into complex 4, the NADH can actually push more H pluses across than, say, the FADH2. And again, those hydrogen ions are pumped in the inner membrane space, so now we have a bunch of those in the inner membrane space. All right, so moving on to complex 2, which is right there, that's where our FADH2 is oxidized to FAD. These electrons and the H plus, again, are transferred to the mobile CoQ and then to complex 3. And so FAD plus produces 4H plus and 2 electrons. Again, the energy from the electrons is what's pushing those 4H plus into the inner membrane space. So again, the main things to know for this is the FADH2, it gives the electrons to that complex 2. And then once those electrons are get moved into that complex 3, it pushes those hydrogen ions through the complex. All right, so at complex 3, these hydrogen ions from FADH2 are pumped into the inner membrane space, as well as some hydrogen ions that are floating around because the electron from the NADH is also floating around. And then once the hydrogen ions are pumped through, electrons are passed to that cytochrome C, and we won't really talk about it, and then it's moved into complex 4. And at complex 4, you get even more hydrogen ions pumped across. Plus, you also add them to water. So you reduce oxygen to make water, which we'll talk about now. So in complex 4, the electrons are used to reduce oxygen to water. So again, the electrons add to water. You also gain those H pluses to make the H2O. And that's what goes on at complex 4. So again, so essentially, all right, we'll talk about this. Yeah, let me talk about it now. So in the summary of the electron transport chain, the NADH is oxidized to NAD+. The FADH2 is oxidized to FAD. And those electrons are used to drive the hydrogen ions into the inner membrane space. And so they first go to complex 1 if they're coming from NADH2. They go down to push more electrons through. Then they ultimately end up at complex 4, where they combine with water and O2 and H plus to end up making water. So the same thing with FADH2. It loses its electrons. They go through the system. It doesn't pump hydrogens in complex 1, just complex 3. And then those electrons go down again to combine with oxygen and H plus to make water. So again, the resulting H pluses are pumped into the inner membrane space, and so it forms this gradient of H plus. So because you form this gradient of H plus, there's this idea of a chemiosmotic model. So again, complexes 1, 3, and 4, they push the protons out of the matrix into the inner membrane space using the energy from the electrons. And so this lowers the pH, but more importantly, it creates this proton gradient, and it's called an electrochemical gradient. So it's called electrochemical because not only is there lots of protons in the inner membrane space and not a lot of protons in the mitochondrial matrix, but also because all these protons are charged. So not only is there a chemical gradient, there's also an electrical gradient. And so all else equal, these molecules would like to kind of even out the charges and the distribution between the two places, between the inner membrane space and the mitochondrial matrix. And so what happens is, because there's this high gradient of H pluses on one side, these molecules will flow back into the mitochondrial matrix, but they don't flow back through anything. They flow through back this complex 5, which is known as ATP synthase. So because you have this big gradient of H+, they want to naturally just go back into that mitochondrial matrix, but the only place they really fit through is through ATP synthase. And so just like a water flows through a turbine in a dam or something like that, these H+ flow through this ATP synthase, and the energy of the H+ is flowing attach a phosphate onto ADP to make ATP. So this ATP synthase, the protons flow back through the channel in the ATP synthase, and this generates the energy to add a phosphate to ADP. And so this whole process is known as oxidative phosphorylation. And there's a little video if you would like to watch it. It's actually kind of cool, but it's kind of hard to show on my slideshow right now. All right, so just kind of a summary again. The citric acid cycle is happening in the matrix of the mitochondria. You produce the NADH as well as our FADH2, which isn't there. But the NADH gets oxidized. It gives up its electrons to complex 1. It pumps hydrogen ions through complex 1. It pumps hydrogen ions through complex 3. It pumps hydrogen ions through complex 4 as the electron makes its way down. And after complex 4, it gets these electrons get added to water and H+. Sorry, keep saying that. They get added to oxygen and H+ to make water. And that's pretty much it. And then so once you have this big concentration of H+ up here, it naturally wants to flow through ATP synthase. And just like a water wheel will turn if water flows through it, the ATP synthase turns, and that energy provided by the H+ flowing allows the phosphate to get attached to ADP. All right, so I think I'll leave that there. So this is, again, we talked about this at the beginning, so I'm not going to make a big deal about it. But this is just kind of an overview of the whole process if you want to look at it again. All right, so we're going to do a little bit of math. It's not too complicated. So the ATP generated from the NADH and the FADH2, it makes certain amounts of ATP because the electrons enter at certain places and the electrons have different amount of energy. The NAD plus enters the electron transport chain at complex 1. And that's a more higher energy state. And so because those electrons have more energy, its energy can be used to synthesize 2.5 ATP. So they just run some math out. We're not going to go through how it does it, but for every 1 NADH, you end up making 2.5 ATP. Because FADH2 only enters at complex 2, the complex 1 can't use its electron to pump through complex 1, just complex 3 and 4. Its energy is only able to synthesize 1.5 ATP. And so that's, again, because the complex 2 is at a lower energy than that complex 1. So again, overall, because you'll need to know this, the NADH, when it gets oxidized to NAD+, using the electron transport chain, it makes 2.5 ATP per NAD+. And the FADH2 goes to FAD plus, and it only makes 1.5 ATP, again, because it enters the electron transport chain at a lower energy stage. So you want to know those numbers because they'll be important. We'll talk about the regulation briefly. Again, if you have high levels of ADP, phosphate, O2, or NADH, it activates it. It's inhibited by high levels of ATP because if you already have ATP. Again, it's strongly dependent on these levels of ADP and ATP for synthesis. Again, if you have lots of ATP, you don't need to make more, but if you don't have any, it'll kind of push into overdrive so you can end up making more. All right, so we're just going to kind of do a final energy tab at the very end. So if you remember, we're going to get energy from glycolysis, when we make acetyl-CoA, when we do the citric acid cycle, and finally when we add those NADHs and FADH2s into the electron transport chain. And so on the next slide, we'll kind of tabulate it up. There's this thing called the malonite aspartate shuttle. We're not going to go into it. It's just a way for NADH to get through the mitochondria membrane. All right, so if we add everything up in glycolysis, you have two NADHs. Each NADH gives you out 2.5 ATP once it gets into the electron transport chain. You made 2 ATP per glucose molecule in glycolysis, 2 net ATP. So total from glycolysis, you make 7 ATP. When pyruvate gets converted into the acetyl-CoA, you make 2 NADHs. And again, this is per glucose. So each pyruvate just gives you 1 NADH. But because you got 2 pyruvates from each glucose, now you have 2 acetyl-CoA. So you multiply that 2 by 2.5 and you end up getting 5. If you remember in the citric acid cycle, we made per glucose molecule, again, you make 2 NADHs here, 2 NADHs here, 2 FADH2s, and 2 NADHs. If you multiply the NADHs by 2.5 and the FADH2 by 1.5, and you sum that together, you get 20. And then if you sum the 7, the 5, and the 20, you end up making 32 ATP. So that is the overall yield of ATP per molecule of glucose that broken down. Again, some of that ATP comes from direct phosphorylation in glycolysis, or that one that we made in the citric acid cycle. But again, you'll see most of it actually comes from the NADH and the FADH2 that you made. So now you can do all the math and sum it up and see where it goes. All right, so that's it for chapter 23. Again, if you have any questions about anything, please don't hesitate to reach out and ask. You can always email me. But again, if you have any questions,